Thank you. Thank you for this nice organization of seminars and the second one of it. So, hi everyone. This is Emre Keskin from Ankara University Evolutionary Genetics Laboratory. Let me give you a brief information about myself. I'm an evolutionary geneticist at Ankara University Evolutionary Genetics Laboratory, EGL, and one of the working group leaders of the ENACONET. I did my PhD on biotechnology. After completing my PhD, I joined many European Union scientific networks and worked in the development of environmental DNA metabarcoding conceptual framework with the task to identify gold standard genomic tools and novel ecogenomic indices and matrices for routine applications for biodiversity assessments and biomonitoring of mainly European water bodies. I mainly focus on eDNA, environmental DNA, metabarcoding of aquatic ecosystems, especially targeting invasive, endangered, and endemic species. And my research group is working on vertebrates, invertebrates, plants, and microorganisms in terms of epigenetics, bioinformatics, and metagenomics. Today, I'm going to talk about main aspects of metabarcoding workflow based on field, laboratory, and bioinformatics. And if I have any time left, I would like to discuss some of our studies in terms of these aspects for real-life experiments and experiences. As I can see from the webinar program, today is 11 hours of lectures, so you already have perfect textbook lectures. So I modified mine into a little bit more a review of suggestions when you are dealing with metabarcoding, kind of a take to lab and apply recipes for you if you are ever interested in metabarcoding and how to design your field, laboratory, and bioinformatics studies. So these will be like a take to lab experiments. So I will do it in 3 parts. Each will be about 40-45 minutes, and I will give breaks at the end of each one. So we can start with the general concepts of biodiversity and how to sample in the environment. As you all know, the anthropogenic impacts cause loss of biodiversity in all habitats, especially in inland waters, and as a result, significant change in the ecosystem. For these reasons, it is necessary to utilize faster research methods in order to develop conservation and management strategies and to detect fluctuations on the biodiversity. In line with the protection of living things, species, and ecological approaches, advances in molecular biology-based methods have resulted in the rapid development of methods for obtaining genetic material, such as hair, skin, feces, and other biological materials left in the environment they live in, rather than the living organism itself. So when the most widely used methods among these examined, environmental DNA, or we call it eDNA, environmental DNA or eDNA based methods takes the first place. Environmental DNA method is a relatively new approach used to monitor the distribution of species. Using this method, it is possible to detect species without actually seeing or capturing them. This method uses DNA-based molecular identification, also called barcodes, to detect types of genetic material from extracellular DNA or cell debris left by the species in the environment. There are many studies showing that the eDNA breaks down at varying times over a few days to a month in the various environments such as water, air, or soil. So therefore, detecting the DNA of a species in water can be used to confirm the recent existence of that species. In other environments, such as sediment, eDNA's persistence can take hundreds of thousands of years or even longer under certain conditions. It is more difficult to confirm the recent existence of an eDNA-based species in these environments like sediments and soils. So environmental factors such as water, endonucleases or ultraviolet radiation, bacteria and fungi that affect the breakdown of the DNA itself directly affect the chance of detecting a species using the eDNA method. When we look at the eDNA, its ability to rapidly and sensitively detect the presence of a target species through eDNA analysis has enabled a wide range of scientific discoveries and technical advancements. For example, isolation of eDNA from ice cores revealed that Greenland was forested almost 2 million years more recently than it is previously estimated. Again, analysis of mitochondrial DNA targets in surface water enabled researchers to distinguish the source of fecal contamination. Environmental testing of cooling towers or other water systems enabled more sensitive detection of Legionella, as you all know, a pathogen that can cause severe illness in the elderly. Or we can give more recent examples such as nowadays we can detect the existence of coronaviruses using samples taken from wastewater treatment systems of the municipalities of big cities to see the relative abundance. Identifying the target species from environmental samples using eDNA has the potential to conduct scientific studies without harming aquatic species and habitats, thus creating an important solution regarding the ecology and protection of aquatic species. eDNA-based methods have the potential to greatly increase scientific data on protected rare or endangered species. It also allows the detection of invasive species while still having low density without establishing a settled population. Apart from those studies aimed at target species, it can also be used to estimate the overall biodiversity of the sampled environment. Studies conducted in the last 2 decades reveal that microorganism communities can be identified at species level by environmental samples taken from water samples and sediment samples. You can see here on the screen the detection sensitivity. It's a schematic representation of the identification sensitivity of the eDNA method compared to conventional techniques like using some kind of nets or photo imaging or other conventional techniques that are already has been used for many years in terms of detecting the biodiversity. So I would like to give you some terminology knowledge related to things about environmental sampling and metabarcoding. So of course, we start with the environmental DNA. It is the DNA extracted from an environmental sample without isolating any target organism. The source of the DNA can be feces, mucus, skin cells, organelles, gametes, or even extracellular DNA. So the eDNA can be sampled from present-day environments like seawater or freshwater, soil or air, or the ancient environments such as the sediment or ice or from permafrosts. Another thing is the community DNA, which is always getting mixed up with the other terms like the macroorganism environmental DNA or the environmental DNA. The community DNA is the mixed mass of organisms. Usually it's indicating the macroinvertebrates for DNA extraction separated from the environmental sample, like from sediment or water. So you separate them first and then you use them as bulk of organisms. And macroorganism environmental DNA is used as environmental DNA originating from the animals and higher plants. So this is the main difference between them. You know barcoding, most of you know the barcoding. The term refers to the performance of DNA sequence analysis using the relevant descriptive barcoding markers such as CO1 for animals, RBCL for plants, ITS for fungi, in terms of taxonomic identification of the species. Metabarcoding term is the taxonomic identification of multiple species extracted from a mixed sample like community DNA or environmental DNA and replicated by or amplified by polymerase chain reaction PCR and sequenced on a high throughput sequencing platform like the Illumina or Ion Torrent or Nanopore, whatever you use. So high throughput sequencing is another thing that is already mixed up with next generation sequencing. Next generation sequencing is a term used for different generations of high throughput sequencing and even Sanger sequencing technologies. So high throughput sequencing is the general term for them. Sequencing techniques that allow simultaneous analysis of millions of sequences compared to the Sanger sequencing method, where sequencing is performed one at a time. Instead, one at a time, in high throughput sequencing, you are sequencing millions of sequences simultaneously. So community DNA metabarcoding is high throughput sequencing using the DNA extracted from the samples, the individuals, assembled or whole organisms after separation from environmental samples such as the sediment or water. The molecular operational taxonomic unit, it's the MOTU or MOTUs. They are the groups identified using clustering algorithms and the predefined DNA sequence percentage similarity. So when I use OTUs or O2s during my seminars, you can understand they are all molecular operational taxonomic units. So in terms of community DNA, it's important to discuss it. I think there are distinct differences between the often confused macroorganism eDNA sampling and community DNA sampling. With the community DNA sampling of animals and plants, targeted groups are usually obtained in mixed form, like by sediments, traps, or meshes, and individuals are extracted from other sample residues and brought together prior to a bulk DNA extraction. In contrast, in macroorganism eDNA sampling, extraction is performed directly from an environmental sample without the process of separating individual organisms or plant material from the sample prior to extraction, and the organism is assumed not to be intact in the sample. Of course, the community DNA samples may contain DNA like the gut contents or intracellular or extracellular DNA. They may contain from tissue, cell, and organellar parts of other organisms. You might never know it. So similarly, samples of macroorganisms eDNA may unknowingly include all microscopic non-target organisms like the protists and bacteria. Thus, the distinction between the two sample types may be partially distorted during the implementation phase. So another important distinction between the community DNA and macroorganism eDNA is that sequences produced from community DNA metabarcoding can be taxonomically verified when samples are not destroyed during extraction. So all the arrays can be created from vouchers using Sanger sequencing. Since the samples for eDNA metabarcoding do not include all organisms or all parts of the organism, such an in situ comparison cannot be performed. You won't have the opportunity to confirm them taxonomically. Therefore, the taxonomic similarities are only those derived from sequences obtained from NCBI Gene Bank nucleotide database, BOLT, or DNA sequences obtained by Sanger sequencing and the local database that you have built yourself. It can be obtained by direct comparison with taxonomically validated sequences, such as local reference databases. In order to at least partially verify the resulting taxon list, the results obtained are compared with the results obtained by concurrently conducted physical, acoustic, or visual-based research methods, or with data obtained from previously created records or literature information for the relevant location. If we look at the scale of the inference levels in eDNA, for the reasons that I explained previously, the difference in source material between community DNA and eDNA has different implications for interpreting the scale of inference for time and space about detected biodiversity. From the community DNA, it is clear that certain species were found at the time and place, but for eDNA, the organism producing the DNA was transported upstream from the sampled location, while DNA was transported from the feces of a more mobile predator species. For example, birds carrying fish eDNA may have been transported, or DNA that was previously present but no longer found in the community, and it might have been released years ago. This means that when using the existence of species in the community based on eDNA data, the scale of inference in both space and time must be carefully considered. While the rapid advancement of eDNA-based conservation practices and their potential contribution to research conducted in this field are exciting, the capture and identification of eDNA and the interpretation of the results of these efforts will more fully explain the ecology of eDNA, such as its origin, its structure, transport properties, and the state, or what we call the fate, of the eDNA molecules. So research from many fields, including microbial eDNA detection, microbiology, using fecal indicator bacteria, and water quality monitoring can contribute to advances in our understanding of the ecology of eDNA. So with ecology of eDNA, I outline 4 main questions aimed at understanding the ecology of eDNA. So they are origin, structure, transport, and state of the eDNA. So here you can see the processes and the features within the 4 main areas of eDNA ecology from A to D. And the key technical challenges you can see in the middle on E part. If we start with the origin part, by origin, it is the physiological sources of eDNA. The physiological origins of the material collected as eDNA remain unclear. Although eDNA applications are rapidly gaining popularity and have become a method used in many studies, microbial studies leading to the detection of genetic material in environmental samples indicate that the eDNA exists in both intracellular and extracellular forms. It is thought that multicellular organisms pour the genetic material into their environment, primarily as tissues and whole cells. And then these cells shed and release the DNA into the environment. Feces remain a possible eDNA source for a wide variety of taxa targeted in aquatic and terrestrial environments, such as amphibians and fish. It is shown that the other body fluids are also a source of eDNA behind the success of detecting the taxa that form thin, slippery mucus layers. Still other studies, there are many studies which shows that in some cases dead carcasses and predator feces can also be used as an eDNA source. Although some studies have examined the size distribution of eDNA behind the size distribution of eDNA bearing particles or shedding particles in aquatic environments to provide clues about their possible origins, any microscopy-based examination or research has no conclusion regarding the content of the collected eDNA. So although the physical identity of the eDNA containing material has not been studied relatively, studies have shown that many factors can affect the amount of genetic material that organisms release into their environment. For example, in an aquarium study conducted on this giant Idaho giant salamanders, it was observed that the amount of eDNA production was positively correlated with the biomass of the species. Another study, a bluefin sunfish, also shows a similar eDNA production and biomass positive relationship. However, when the results are discussed in depth, it reveals that the juvenile sunfish DNA production and release rates are higher than adults. So in terms of target amplicon copies per hour per gram fish weight. So ontogenic factors such as changes in behavior and metabolism affect the DNA production. In another study, researchers found that the increased feeding behavior of the carp species increased the eDNA release rates. So if you are making your sampling in a season like early May, the eDNA abundance will give you more eDNA copies. But if you are doing in winter time where the carps are not feeding that much and the eDNA abundance will be lower. So they release less amount of eDNA. Or if you sample during their spawning season, there will be lots more eDNA in the environment than you will expect. So relative abundance studies should be modified or optimized accordingly. When discussing the main research questions facing the future of eDNA studies, it must be kept in mind that local abiotic and biotic environmental conditions may affect the eDNA production. For example, a study demonstrated that temperature and microbial activity contribute to the DNA release of plant materials in freshwater sediments. It is important the understanding the source of the genetic material an organism emits it into the environment and the factors affecting eDNA production will definitely improve our understanding of the taxa and environments in which eDNA would represent an effective conservation and research tool. As an example, the more knowledge to be gained about the relationship between eDNA production versus the organism size, age, and biological activity may guide how eDNA sampling should be done. Conservation studies using eDNA can maximize sampling success by taking into account temporal and spatial patterns to target the sampling periods in which genetic material accumulates. On the other hand, ecological knowledge of the relative differences between different species in eDNA-defined populations or between different age classes in a species will allow the method to be used more precisely and accurately for research and conservation studies despite all its difficulties. On the screen, you can see the difficulties in abundance estimation in eDNA metabarcoding studies, the subsampling, primer bias, and both the primer bias and subsampling always give different results than expectation. You can see, according to the prior knowledge on the ecology of this lake, the expectation of the results should be like this, but because of subsampling and during the DNA lost through the subsampling process, it might be underestimated. Or because of the primer bias, some species may be overestimated, or because the joint effects of primer bias and subsampling, most of them are underestimated, but some of them are overestimated than expected. So it's a matter of advantages and disadvantages. You need to know the ecology that you are working. So the ecology of eDNA origin is something, and the state is another thing that we need to consider. eDNA forms in nature are called the state or the status of eDNA. One of the most important questions about the origin of eDNA is its situation in the environment. Based on the results of previous scientific studies, it is known that eDNA exists in both intracellular and extracellular forms. Probably eDNA is transitioning from the intracellular state to extracellular state over time, and there is sufficient knowledge about the various ecological processes that accelerate this transition. The application of eDNA metabarcoding methods to different sample types taken together provides the opportunity to make an assessment specific to the results obtained over time, local extinctions, and long periods of time frames. You can see the time scale and the geographical state here. In terms of time scale, you can see how the eDNA status change from different geographical scales. Environmental DNA sample types have different spatial and temporal scopes in different habitats. Until recently, there is no such a systematic comparison between eDNA sampling methods, and so far, trial and error and logistical restrictions have been effective in determining the method in this area. Understanding the state of the eDNA, will undoubtedly provide an important perspective for obtaining and analyzing it. For example, using this information to be obtained, certain eDNA particles can be targeted more specifically using certain filter size and capture methods, as you can see in here. So, moreover, a better understanding of the size distribution of not only target eDNA particles, but also non-target eDNA and other particles responsible for PCR interactions or inhibition may lead to the use of methods such as multi-step filtrations or prefiltrations. The knowledge of size distribution of various particles in the aquatic environment will provide us with a preliminary knowledge of the balance between the filter pore size and the amount of water that can be filtered before the filter clogging. Smaller filter pore size capture more particles, but limit the filtration volume and the filtration speed. Larger filter pore sizes filter larger volumes of water faster, but may allow smaller target particles to pass through. A more comprehensive knowledge of the size distribution of target and non-target particles will allow the selection of filter size and sampling strategies that best guide sampling with precision and with more efficiency. Another issue about the ecology of eDNA is the transport part. The transport is defining the physical movement of eDNA in the environment. After shedding from an organism, eDNA may move in its environment and affecting the results obtained from eDNA-based studies. So you might get along with an unexpected DNA or OTU, which shouldn't be there. In a study on the downstream motion of potential DNA, eDNA, they produced synthetic DNA tracers for an injection tracing experiment in 2 streams. I think it was in Netherlands. So with the synthetic DNA tracers, they put it inside the water. They know the synthetic DNA sequence. So from different points, they take water samples and to see how the DNA is transported. So observing that the physical movement of the synthetic DNA tracers conformed speed of the standard tracers, the researchers demonstrated a significant decrease in the amount of eDNA after the downstream movement, and experimentally demonstrated the existence of adsorption, biological uptake and other interactions between DNA molecules and the environment where the flow occurs. Despite these interactions, synthetic DNA remained detectable even at a distance of, I think it's 1,192 meters. So it's the end of the lane. And when they release the DNA from the beginning and when they take samples from the downstream, they can detect the DNA, but there is some fluctuations in the amount of it. So this is something that should be also considered. As in the example of synthetic DNA tracers, the extracellular DNA is the only one component of DNA detected in eDNA-based conservation and research applications. eDNA is thought to represent a complex mixture of particles ranging from extracellular DNA molecules to whole cells and the aggregation of cells. Recently, studies have started to examine how this complex DNA signal is transported in aquatic environments or other environments like the sediments or air. For example, researchers using the eDNA method to detect invertebrate species they have observed horizontal movement of the eDNA within the water column, and when they calculated this, it's more than 12 kilometers, more than 12 kilometers, something like 13 kilometers. So even at 13 kilometers at the other end of the downstream, they can still detect the DNA released from the invertebrates. Also, studies on vertical movement of fishy DNA in sediment and its accumulation in lake and river sediments show that the resuspension of eDNA in sediment is an important element of eDNA ecology. In general, understanding this transport rates the detected eDNA to the presence of the species in both spatially and temporally is important. And the last status of the DNA is the structure or the fate or the fate of the eDNA. It's the persistence of eDNA in the environment. DNA has a very long time-limited chemical stability under certain conditions. As you know, in most cases, eDNA begins to degrade as soon as it's shed from an organism. So factors affecting the continuity of eDNA in environments are the focus of recent studies that take into account studies in a wide variety of disciplines. These studies concluded that factors affecting eDNA persistence fall into 3 broad categories. One of them is DNA properties, which we can classify them like conformation, length, cellular and organelle based, relationship to membrane. These are all the DNA properties. In terms of abiotic environments like light, oxygen, pH, salinity, abundance, and even composition of the substrates. The third category is the biotic environment, which we can classify the composition and the activity of microbial community, and the extracellular enzymes can be also included into that. So as a result that can be derived from these properties, eDNA ecology affects population inferences. In terms of A, the origin, and eDNA from reproduction and the composition can produce similar temporal patterns despite different origins. In terms of the state, the B part, different types of filters may yield different concentrations of eDNA that reflect particle size classes rather than population size differences. In terms of transport, the resuspension of old sedimentary eDNA can produce false variation inferences after organisms are gone. And in terms of fate or the status of the eDNA at D, different environmental rates of eDNA degradation may affect inferences regarding population size or biomass from eDNA concentration. So there is something important that I need to also show to you is the minimum information, the MI, it's called MI, for analysis of eDNA samples. The general recognition of the importance of details regarding the experience gained from the previous studies has led to the creation of many minimum information guidelines that are currently reinforced through the MIBBI project. So for this purpose, an MI guide was created for the analysis of eDNA samples obtained from environment. And these are in addition to the standard requirements for publication of molecular analysis results. Including this level of detail in published studies will improve the quality of the results from studies in this area by increasing the communication about techniques and quality controls. So you can visit this website and you can see the minimum requirements for an eDNA or DNA metabarcoding study should include in terms of design of sample collection, sample preservation, extraction process, probe-based qPCR requirements, and high throughput sequencing requirements. In terms of designing the sample collection, the presence, absence, and quantity is important. Contamination precautions, including negative controls, collection volume, container material, replicates, depth, size, the sampling site descriptions are important. For sample preservation, the method, temperature, duration, filter type, filtering location are also matters. During the extraction process, the contamination precautions, negative controls, the kit protocol adjustments are important for a better publication. And when you are designing a probe-based qPCR, the design and validation methods, primer probe sequences, amplicon lengths, positive and negative controls, inhibition detection, thermal profiles are all important details that you need to share with the scientific community. Also, in terms of the high throughput sequencing, the library type, enrichment strategy, library preparation protocol, the platform that you used, read length, read pairing, expected fragment size, primers, sequencing adapters, they are all important. I will give more details about them in the following lectures. So despite the advantages and provided by the environmental DNA technique, the results of metabarcoding studies are affected by a number of sensitivity and attention deficit problems observed in the field, laboratory, and bioinformatics workflows. The eDNA workflow determined after study design on the screen you see basically consists of three main steps: field, laboratory, and the bioinformatic analysis. You can see the important guiding steps to be considered for the design and implementation of the environmental DNA metabarcoding studies. To summarize what's on the screen, the field step involves obtaining a preservation solution or a frozen sample prior to DNA isolation. The laboratory step has four basic steps: DNA filtration, PCR used to replicate the target gene or gene region, indexing, and reading the assembled libraries by high throughput sequencing. The sequence obtained after laboratory processing of samples is processing of sequence information using an optimized bioinformatics workflow in which the information is processed, as you can see here. We will see them in the following parts. To conclude this part, the beginning, I think we have some things to consider before having a break. So, non-capture, non-invasive methods, as we described the environmental DNA sampling. The environmental DNA can be intracellular, extracellular, or it can include cell debris. The relative abundance is an important issue that needs to be optimized according to many factors that we discussed. Community DNA is something different. It's origin, transport, structure, and state of the DNA matters. So you need to know the eDNA ecology very well. And there are some minimum information standards for metabarcoding studies. So you need to walk through them in order to design your study easily publishable. So this will be the introductory part. We will continue with sampling in the field. I will give some tips about how to design your studies in the field. But before that, if there is any questions, I can answer, or we can give a short break. Any questions? Okay, then I think we can give a 15-minute break. Or Meriç, is it okay? Or what's the plan?
Okay, hocam, it's up to you. We can give a break, 15 minutes.
Yeah, we can give a 15 minutes break. So see you at 11:15.
Okay.
And meanwhile, you can, if you have any questions, you can